Cancer. It needs no introduction. Despite all our advances in modern medicine, healthcare, and pharmaceuticals, it continues to plague our world and affect millions of lives, with one of the most prevalent types being breast cancer. Don't take my word for it. Even the WHO dictates that nearly 2.3 million people are diagnosed with breast cancer every year, and nearly 700,000 people die annually. Now, you may have heard about emerging treatments for cancer patients, but these treatments have been relatively unhelpful in addressing breast cancer specifically. Why, you might ask? Well, that's because breast cancer works fundamentally a little bit different than other types of cancers. Cancer, in essence, occurs when the body's immune system cannot find the actual cancerous cells, which have begun multiplying themselves uncontrollably. These emerging treatments usually leverage the body's own immune system to hunt and fight these cancer cells. So this is all well and good, but what can we actually do to stop this? I mean, I'm just a 16-year-old without access to a lab and millions of dollars worth of research funding. So I had almost given up until I found Benchling, which is a modeling software that allows us to create solutions all from the comfort of our home. With Benchling, we can create a workaround that enables us to beat breast cancer once and for all. In this tutorial, I'm going to walk you guys step by step on how I created a novel plasmid design capable of tumor suppression preemptively, meaning that can find and destroy cancer before it ever becomes a concern. With that being said, before I hop into this tutorial, it's important to have a solid understanding of gene expression, specifically through protein transcription and protein cascades. I've actually written a couple of articles on these topics that I've linked down below, so if you need extra help on that, feel free to check them out. And with that all being said, let's begin the tutorial. So after we've created an account on Benchling, we're going to open up a new project, and we can call this project anything, but for today's sake, I'm going to name it the breast cancer plasmid. Then we can import an existing DNA sequence from an online database to serve as a backbone for our plasmid. So to do that, all you have to do is go DNA RNA sequence and import it, and then import from database. The plasmid we're going to be using will contain the muron interferon gene, which is present in mammalian cells, and it encodes for the cytokines, which I mentioned earlier, which are going to be integral in enhancing our body's own capability of fighting breast cancer if it ever begins spreading in the body. I've actually linked this plasmid down below in the description if you guys want to use it. The plasmid has a couple of useful qualities, namely it has several protein promoter sites all around it. You can think of protein promoter sites as sensors almost. They scan for the presence of a certain molecule, and once it gets detected in a significant enough quantity, the prom site activates the gene. Specifically, we're looking for the CMV protein promoter site, as there are several studies citing a correlation between the presence of CMV and cancer. Similarly, we're looking for the SV protein promoter site, as it's activated in the presence of malignant tumors. The plasmid also has neomycin and ampicillin, which are selection markers. If we were creating this plasmid in real life, selection markers would be useful in allowing us to verify our gene is working successfully. However, they don't really serve a purpose in actually addressing the breast cancer and the tumor suppression. So we're going to insert the plasmid into Benchling. So all we need to do is copy and paste the URL and then put it right here, press search, and Benchling will do the rest for us. We don't need to actually touch any of this, so we can just press import. Voila, Benchling will have imported the entire plasmid. Now you'll see this annotation that says source. It's completely redundant, so we can just go ahead and right click and then press delete annotation, and therefore it will be gone. Now you'll see that we don't actually know where the segment encoding for murine interferon is. It's important to understand that the entire plasmid isn't used to encode for murine interferon. You can see here that there's various protein promoter sites along the actual plasmid, as well as various ORI sites, which are mechanisms used for duplicating the plasmid in a lab setting. We don't want to accidentally delete any of the bases responsible for murine interferon, so we have to annotate it, but we don't actually know where to annotate it. So we can go back by going to the plasmid site on add gene, going to the map image of the plasmid, and it shows us that the INF transcription site is between the restriction enzyme NHEI and not I. So we can look for these restriction enzymes on Benchling, and we can highlight the bases between the two enzymes and create an annotation. So see over here, here's the NHEI. There we go, and then we can just keep scrolling for a little bit, and there you go. That's not I. So all you have to do is go up here, see, go right there, and then keep highlighting the bases, not that much. Go back, and there we go. All the way to not I, and then all you have to do is right click and then press create annotations, and we're gonna call it the INF, and we're gonna make it pink. There we go. Save, and there you go. Just like that, we have actually put in the annotation of the INF gene segment on the plasmid itself. So the plasmid is already good at tumor suppression, yet it's not entirely effective. Releasing co-stimulus induces an immune response, but we want to ensure the cell dies for sure. There's no outliers. Occasionally, our immune system will become corrupt and it will actually kill the cell. And that's why every cell has a last resort switch, which is apoptosis, or in other words, cell suicide. However, to activate this, we're going to need the help of another gene, P21. So, following the same procedure as last time, we're going to import a plasmid containing P21. So, here's the tab, again, back on add gene. This is the plasmid, and there you go. You can see it contains P21. And again, I'll be linking this down in the description so you guys can check it out. So, again, following the same thing as last time, we just copy the URL. We import a DNA RNA sequence, from the actual database itself. We press search, and Benchling will do the rest for us. We don't have to touch any of this again like last time, and we just press import. And we'll have the actual plasmid right here. And same thing as last time again, we can delete the source annotation as it's unnecessary. So we're going to be highlighting the P21 gene here as well as the HGH polyA signal right down here. The polyA signal, also known as the polydactyl signal, is the ternary sequence. So in other words, this tells the RNA polymerase to stop transcribing the gene. Because note that one gene can encode for multiple proteins, which is why a ternary sequence is necessary. So we're going to go ahead and just highlight this, make sure we don't miss any base pairs. There you go. But we can't just copy paste this into our murine interferon gene. We actually have to perform a restriction enzyme digest, which in essence is when you use an enzyme to make a cut into a plasmid and then insert a new piece of DNA in. So to do this, we go back to our original gene, which is the murine interferon, and we can open up the linear map, which shows, again, the whole plasmid, but just in linear format. And this is the INF gene right here, and these are the promoter sites. So that's the T7 promoter site, and this is the T3 promoter site. Again, reinforcing the whole idea how promoter sites are sensors for genes to be activated. And if you're wondering why there's two promoter sites on either side of the gene, it's because plasmid DNA is actually circular. So transcription can occur in either direction, so it's important to have promoter sites in both the 3' and the 5' direction. So if we were to just zoom in here, because there's a gap between both promoter sites, you can see there's two enzymes right here that would cut right after the gene. So we don't want to cut in between, which is why we might not use an enzyme such as NRUI, because it will be cutting right in the middle. We can use NOT-I and EAGI. So first we have to find out what the actual ideal enzyme is, so we can double click on both enzymes. And you can see this enzyme creates two cuts, which we don't want to do because then we have to worry about reassembling the plasmid back in, and that becomes so tedious. So we're going to try NOT-I. And there you go, NOT-I would be ideal in this case because it only makes one cut, and that's all we want to do. We just want to make a nick instead of cut the actual DNA entirely off. So before I continue, you'll notice there's a black jagged line that occurs when I hover over the NOT-I enzyme, and this actually denotes where exactly the enzyme will cut, which in our case creates a sticky end. So to simulate a restriction enzyme digest, all we have to do is actually highlight the recognition site, which would be from here to here, I believe. Nope, we missed a base pair. There we go. And we actually have to go ahead and delete the recognition site. In real life, the enzyme would be doing this, but in Benchling, we have to manually go ahead and do this. And you'll notice we're actually cutting off a couple of bases at the end of the INF gene, and that's actually not to worry because our body has self-repair mechanisms such as DNA polymerase, which can fix the single base deletion caused by our enzyme. So we can just go ahead and press delete. It'll ask us to confirm whether or not we're going to delete this, and yeah, we do want to delete it. So there we go. And then now what we do actually we go back to our P21 plasmid, where we've actually highlighted the P21 gene as well as the polydactyl signal, and we just hit Control C, and it'll say a copy to clipboard, and then we go back to our murine interferon gene, and we just press Control V. There we go, and it's gonna just confirm that we actually do indeed want to copy over the annotations as well as the bases. So we just press Enter, and there you go. So you can now see on the plasmid that we've actually added the P21 segment and as well as the polydactyl segment to our murine interferon gene. We still need to add a start codon that begins translating the DNA. And you probably thought that was the role of the promoters, and you'd be right, but also wrong. See, promoters actually initiate transcription, while start codons initiate translation, with transcription being the synthesis of RNA from a DNA template, while translation is the synthesis of a protein from an mRNA template. There's several start codons that we can use, which are also otherwise known as Kozak sequences, ranging in application and effectiveness, but the one that we're going to be using is the one shown on screen above you, and I've also put down in the description below. More specifically, we have to insert this Kozak sequence corresponding to the P21 gene in front of P21, as the INF gene already has a different Kozak sequence integrated into this. So to do this, again, we have to perform a restriction enzyme digest. So we have to put it before the P21, so we could do it on either four of these enzymes, but obviously we don't want to cut that much off of the INF gene, so we're going to be looking at these two ACI and SALI. So we can check how many cuts. So ACI creates one cut, and then SALI also creates one cut. So we don't have to use either one of these, but we're going to use ACI simply because it creates a 2-base overhang, while SALI creates a 4-base overhang. And having a 2-base overhang usually is much easier to work with in the lab. So again, same procedure as last time. And all we have to do is actually just copy paste the strand into the DNA. So there we go. And Benchling will automatically fill the complementary strand out. And just so we make sure we don't actually lose this segment of DNA in the rest of the annotations, we're going to actually annotate this. So we're going to create an annotation called a Kozak sequence. And we're going to make it light blue. And there we go. So where we have the Kozak sequence for the P21 gene. And lastly, before we end off, we want to rename our plasmid to make sure we don't confuse it with our other plasmids. So we can go over here and rename. I think that's a fitting name. And there we go, we're done. Congrats, you successfully created a plasmid capable of tumor suppression. The way it works is that when SV40 or CMV are detected in the body, it will begin cancer suppression via the P21 pathway. The thing is, our body already contains a copy of P21 in its genome, so this will serve as almost a backup of sorts. This is because usually as the older we get, the copy of P21 inside of our body gets mutated and thus more ineffective. Additionally, the interferon will encode for the cytokines which I mentioned earlier, which will initiate an immune response and maximize the effectiveness of our plasmid, ensuring the cancer dies and does not metastasize. Be sure to check out the second video I have in this series where I create a guide RNA for a plasmid. It's been a pleasure working with you all, and this is Krish out.